today's talk is endothelial NMDA receptors mediate astrocyte-induced cortical vasodilation.
All right, good afternoon, everyone. My name is Adam Hogan-Cann. I'm a Ph.D. student at the University of Manitoba in Chris Anderson's lab. And I'm really excited to be here today, have the opportunity to speak with you about some of the work we've been doing recently, where we've been looking at the role of endothelial NMDA receptors in astrocyte-mediated cortical vasodilation. So an important regulatory mechanism that controls brain blood flow is functional hyperemia. Just briefly, this is characterized by rapid increases in blood flow in response to elevated neuronal activity. And a key driving force of functional hyperemia is neurovascular coupling. And this is really characterized by a complex communication between cells of the neurovascular unit in response to increased synaptic activity. And we know that astrocytes have emerged as key mediators and important regulators in neurovascular coupling. And it's been shown that they can respond to synaptic activity, and they relay these signals between actin neurons and the local vasculature. Now, interestingly, the cerebrovascular endothelium also seems to be critically important for whole brain conducted vasodilation for functional hyperemia. But what's really needs to be determined is how these vasodilatory signals from the brain are reaching the cerebrovascular endothelium to initiate these vascular responses. So our lab hypothesized the main objective for the study was to determine whether or not vasodilatory astrocytic signals can reach and directly communicate with the cerebrovascular endothelium to initiate local vasodilation. And we hypothesized that this is dependent upon endothelial nitric oxide synthase and activation of endothelial NMDA receptors. So there are three key pieces of evidence to support this hypothesis. So firstly, work that was done previously in our lab where we took isolated brain arteries that were denervated, and we showed that these arteries are capable of dilating in response to NMDA receptor coagonists, specifically glutamate and D-serine. Importantly, this dilation was dependent upon intact endothelium and could be blocked by eNOS inhibition. As well, this dilation was sensitive to NMDA receptor antagonism. We've had consistent results in a brain slice model where we've shown that independent and individual activation of a single astrocyte results in local vasodilation of a penetrating arteriole, and that this is also dependent upon NMDA receptor coagonists, glutamate and D-serine, and required, again, eNOS activity. And as well, our group and others have demonstrated that brain endothelial cells express the critical NMDA receptor subunit GLUN1, which suggests that NMDA receptors are present in these cell types. So collectively, these pieces of data suggest that NMDA receptors expressed on the brain endothelium may be capable of mediating local vasodilation. So while we're hypothesizing that an endothelial NMDA receptor is an important mediator of hyperemia, what's really needs to be first looked at is the expression profile in the cell type, as this hasn't been fully studied in the past. So using whole cell lysates from primary brain endothelial cells cultured from mice, we employed a co-immunoprecipitation approach in which we targeted the critical GLUN1 NMDA receptor subunit. And using this immunoprecipitation approach, we identified GLUN2C co-immunoactivity, which suggests that GLUN1 and GLUN2C subunits are directly associated. And this result was confirmed in B using the reciprocal approach, in which GLUN2C was immunoprecipitated as well. So these suggest that brain endothelial cells express NMDA receptors composed of both GLUN1 and GLUN2C subunits. So if we're hypothesizing that these receptors are important mediators of hyperemia in the brain, then we would expect that these are located on the abdominal or the brain-facing side of the cerebral vasculature. In this orientation, they are configured so that they are capable of receiving vasodilatory signals from the brain. So to investigate subcellular distribution of endothelial NMDA receptors in situ, we employed immunoelectron microscopy, again targeting the critical GLUN1 subunit. So we took transverse sections of mouse brain, and this allowed us to have cross sections of penetrating arterioles and cortical vessels. And as you can see in figure C, there was a clustered labeling of GLUN1 immunogold beads localized primarily along the abdominal or the brain-facing endothelial membrane. And this is highlighted by the purple arrows clustered around the yellow line, the yellow trace, which is the abdominal membrane. In fact, just over 80% of the total endothelial GLUN1 membrane labeling was localized along the abdominal membrane. And this was consistent across all vessels that were sampled within the cortex. So this is an important piece of evidence as it demonstrates that endothelial NMDA receptors have membrane polarity and shows that they're in a configuration suitable for receiving dilatory signals from the brain. And this is an important piece of evidence to support our hypothesis that they are involved in brain academia. So we wanted then to look at whether NMDA receptor subunit expression is correlated with functional NMDA receptor activity. So to do this, we looked at nitric oxide production in brain endothelial cells in response to NMDA receptor activation. So primary brain endothelial cells were loaded with the fluorescent nitric oxide indicator, DAPFM, and that's shown here in response to glutamate in combination with D-serine, but not on its own, significantly enhanced nitric oxide production. We directly assessed the involvement of the NMDA receptor in this process, and this was done by pre-treating endothelial cells with NMDA receptor antagonists. So CNA, pre-treatment with either AP5 or DCKA, which are blocking the glutamate or D-serine binding site respectively, completely eliminated the observed production of nitric oxide. We next wanted to assess the involvement of calcium in this process. So this was done by re-treating endothelial cells with either MK1 to block the NMDA receptor channel 4, or by pre-treating with BAFTA-AM, which is an intracellular calcium chelator, or finally by removing calcium from the buffer. So shown in B, MK1 blocked calcium through the channel 4, and this was associated with a significant decrease in nitric oxide production in response to glutamate and D-serine. Similarly in C, we found that chelation of intracellular calcium and removal of calcium from the buffer also eliminated the observed nitric oxide production. Thus, the results from figures B and C are consistent with an effect dependent upon influx of calcium. And lastly, in figure D, we demonstrated that this production of nitric oxide is dependent upon eNOS activity. And this was done by pre-treating endothelial cells with a eNOS-specific inhibitor, LNIO. So just to summarize this slide, which is a little busy, we've shown that glutamate and D-serine co-treatments increases nitric oxide in brain endothelial cells, and that this nitric oxide response is dependent upon activation of NMDA receptors, calcium flux, and eNOS activity. So to better understand the role of an endothelial NMDA receptor in brain hyperemia, we developed an endothelial-specific loss of function mouse model, and this was developed using the CRE-LOX system, in which mice with LOX-P sites flanking critical regions of the GLUN1 gene, brain 1 on the left, is crossed with a mouse that expresses the bacterial enzyme CRE recombinase under control of the endothelial-specific promoter TIE2, shown on right. And the resultant offspring have GLUN1 levels reduced specifically within the endothelial cells, while all other cell types which do not express CRE maintain normal levels of GLUN1. And we validated this mouse model using immunoelectron microscopy. You can see in figure A, when we looked at GLUN1 immunoglobulin labeling in the brain endothelium, there was about a 50% reduction in immunoglobulins in the CRE-positive mice relative to CRE-negative littermate controls. And importantly, the synaptic levels of GLUN1 were unchanged between these mice. And this was in agreement with in vitro data, so brain endothelial cells cultured from CRE-positive animals also displayed about a 50% reduction in GLUN1 immunoactivity relative to CRE-negative cells. So thus, our animal model demonstrates endothelial-specific knockdown of GLUN1, and we hypothesized that if the mouse has reduced levels of endothelial GLUN1 expression, then they might also have impaired endothelial NMDA receptor signaling. So specifically, we were interested in addressing the role of endothelial NMDA receptors in nitric oxide production and vasodilation in situ. So this was done using an acute cortical mouse slice, and we loaded these slices with a fluorescent nitric oxide indicator DAPFM, which is shown in red. The slices were also loaded with a caged calcium compound in combination with a calcium fluorescent indicator, rhodamine-2, which is shown in green. And this was used to label astrocyte calcium. So using two-photon microscopy, we were able to image penetrating cortical arterioles that have direct achievement with perivascular astrocytes. And then by using targeted two-photon flash photolysis, we can photoactivate calcium signaling within a single astrocyte and then monitor production of endothelial nitric oxide and changes in vascular lumen diameter downstream of astrocyte calcium signaling. So you can see in the video, in response to two-photon activation of astrocyte calcium, there's a dramatic increase in astrocyte calcium fluorescence, and this is followed by a slower, more gradual buildup of the red nitric oxide signal. So interestingly, when we looked at the 5-minute accumulation of nitric oxide production in response to two-photon activation, NMDA receptor antagonism, either by AB5 or DCK, shown in B, significantly reduced the production of nitric oxide. Similarly, endothelial-specific GluN1 knockdown, shown in the white bar in B, also significantly reduced the production of nitric oxide in response to astrocyte calcium signaling. Now we know that nitric oxide contributes to vasodilation, and it does so in part by modulating the metabolism of arachidonic acid. And specifically, it's been shown that nitric oxide can inhibit the production of the smooth muscle vasoconstrictor 20 heat, and thus it favors production of more vasodilatory metabolites such as prostaglandins. So we wanted to address whether NMDA receptors are all similarly involved in modulating arachidonic acid metabolism. So we did this, and this is represented by figures C and D, by using, again, an acute cortical slice model, and we back applied glutamate and D-serine, and then analyzed tissue levels of PG2 and 20 heat 5 minutes after using ELISA. And in panel D, you can see that Cre-negative slices in black had an unchanged level of tissue levels of 20 heat. However, the Cre-positive slices that have endothelial-specific GluN1 knockdown had significantly increased levels of 20 heat. In contrast, in C, the black bars represent Cre-negative slices and have a dramatic increase in tissue levels of the vasodilator PGE2 in response to glutamate and D-serine co-treatments, whereas this effect was lost in the Cre-positive slices. So effectively, this slide demonstrates that in response to endothelial NMDA receptor suppression, there is reduced nitric oxide production, and this is associated with altered arachidonic acid metabolism, favoring production of the vasoconstrictor 20 heat. And lastly, the big question of our study was to determine whether endothelial NMDA receptors influence vascular lumen diameter. So shown in panel A by the green line, we looked at the cumulative vasodilatory response after two-photon activation of astrocytes. You can see in green that the Cre-positive slices with, again, endothelial-specific knockdown of GluN1 had a significantly reduced vasodilatory response relative to Cre-negative slices. So this suggests that arterioles with endothelial-specific GluN1 knockdown have reduced vascular reactivity in response to direct astrocyte activation. So in summary, our lab has evidence to support a novel mechanism of neurovascular coupling, and this links astrocyte signaling, endothelial NMDA receptor activation, and eNOS activity. And this helps to provide some information how vasodilatory signals from the brain are coming to reach the cerebrovascular endothelium, and therefore may contribute and play an important role in some of the endothelial-dependent mechanisms that are required for conductive vasodilation needed for whole brain hyperemia. And we confirm the functionality of this pathway with our novel loss of function mouse model, where we associated reduction of endothelial NMDA receptors with reduced nitric oxide production and altered arachidonic acid metabolism. And I'd like to finish by thanking my advisor, Chris Anderson, and also colleagues in the lab, as well as our collaborators at the University of British Columbia and the University of Manitoba, and as well as our various funding sources. And thank you for your attention.
So, is this, I'm just thinking about the NMDA receptor here. Does it have AMPA receptors to lead the voltage block there, or how does it, is it like a metatropic type of NMDA effect?
So something that's not entirely clear at this point. Endothelial cells do express AMPA receptors, but because of identification of the GluN2C subunit, that will likely suggest that they have reduced sensitivity to magnesium blockade, and therefore aren't necessarily requiring that AMPA depolarization to relieve that magnesium blockade.
Thanks.
Yeah.
Quick question. You showed actually that the receptor levels were decreased in synapses using mushroom block. Did you ever look at neuroactivity and to check to see if there's a difference in network activity in neural?
Nothing that we've looked at this point. Our plan is to move in vivo to address this sort of animal model. That's not something we haven't done yet.
Well, thank you very much to all the speakers for wonderful talks. I'm sure if you have any more questions, I'm trying to find the speakers. Thank you all for your attention. This ends the Symposium.